Buddy, it's your friend Daniel here, and welcome back to another tuition video. And in this video, we're going to be going over GCSE Biology 2018, and we're going to be going over the higher paper. This is the first paper properly made by AQA, and without further ado, let's jump straight in. Question 1.1. Two symptoms of infection by Salmonella are vomiting and diarrhea. What causes these symptoms? And what causes the symptoms of the Salmonella bacteria is toxins secreted by the Salmonella. And for 1.2, give 2 ways a person with a mild infection of Salmonella can help prevent the spread of the bacteria to other people. And this is by washing their hands after using their toilet or disinfecting clothes and surfaces. And 1.3, in very serious infections of bacteria, a doctor can prescribe drugs to kill the bacteria. What types of drugs can the doctor prescribe to kill the bacteria? And this is antibiotics. Any bacterial infection, antibiotics can be used against as long as it's not resistant to antibiotics. And you don't use antibiotics on non-bacterial infections and diseases. And for 1.4, a person with AIDS may take longer than a healthy person to recover from a Salmonella infection. And this is due to their immune system being damaged, which prevents white blood cells from effectively killing the Salmonella. Then on to 1.5. Salmonella bacteria can be transferred from chickens to humans. Chickens can be vaccinated to prevent the transmission of Salmonella bacteria to humans. Suggest one other way farmers could prevent the transmission of Salmonella from chickens to humans. And this is by slaughtering the infected chickens. It may seem a bit cruel. AQA doesn't care, they put it in the mark scheme anyway. Next up, we have a little bit of a graphical analysis question. A restaurant owner employed a scientist to test the effectiveness of 2 cleaning liquids. The scientist took samples from 2 work surfaces, before the surfaces had been cleaned with the cleaning liquids and after the surfaces had been cleaned with the cleaning liquids. The samples were then analysed for the number of bacteria they contained, with these results being shown in figure 1. And you can just see the results here, number of bacteria in times 10 to the power 3, cleaning liquid A and cleaning liquid B, and for both the sink and table before and after cleaning. And we're asked which cleaning liquid is more effective. And it's the cleaning liquid that's more effective is cleaning liquid B, because it reduces the bacteria more than cleaning liquid A in both the table and the sink. Now moving on. The scientist investigated the effect of cleaning liquid A and cleaning liquid B on Salmonella bacteria grown in a laboratory. Figure 2 shows the way the investigation was set up. And we have filter papers soaked in water, cleaning liquid A and cleaning liquid B, and Salmonella bacteria growing on agar. The Petri dish was placed in an incubator at 25 degrees Celsius for 48 hours. After 48 hours, the scientists calculated the area around each paper disc where no bacteria was growing. Water had 0 area with no bacteria growing. Cleaning liquid A had 11 centimeters squared, and cleaning liquid B had 13 centimeters squared. And we asked what measurement would the scientists need to calculate the area where no bacteria was growing? Well, you can't measure area directly, so you need to measure the radius where no bacteria is growing, and then use the formula pi r squared. And then we're asked to give one change to the investigation that would allow the scientists to check if the results are repeatable. And what we do is we simply repeat and see if the results are similar and give the same result. This is a bit dumb because what changes could the scientists make to make it repeatable? And the answer is to repeat it. I don't think that's immediately obvious based on the wording of the question, and I just think it's a bit weird for AQA to have the answer repeat the question and just have it say repeat it. But other than that, I think it's a fine question and a bit easy to pick the mark up on. And for question 1.9, the scientists showed results to the restaurant owner. Both cleaning liquids cost the same per decimeter cubed, and were asked to suggest one other factor that the restaurant owner should consider when choosing which cleaning liquid to use. And this is the effect on other pathogens, because they're solely focusing for this one on Salmonella bacteria. Now moving on to question 2. Metabolism is the sum of all kinetic reactions in the cells of the body. One metabolic reaction is the formation of lipids, and we're asked to give one other metabolic reaction besides the formation of lipids, and this is respiration. And we're showing the mean metabolic rates of humans of different ages based on their gender and their age. And we're asked what two conclusions can be made from the data in Table 2. And this is that males have a higher metabolic rate than female after 5 years of age, because at 5 years of age they're the same, and then after that, for males it's higher. And the mean metabolic rate for females decreases faster than males up to 25 years of age. So you can see it decreases a lot faster up to 25 and stays constant, while for men it's constantly decreasing. Then we're asked to calculate the percentage decrease in the mean metabolic rate of males between 5 and 45 years. For 5 years it's 53, and for 45 years it's 46, so we just use the formula they give you, rather than asking them to give you the formula. 53 minus 46 is the decrease. Over 100, it's not over 100, it's over 53. Okay, I still, for some reason, got the answer correct, though. Probably just a silly mistake when I was writing it down, yeah. 53 minus 46 over 53 times 100 equals 32.0754. I'm asked to give it to three significant figures, which is 32.1. Then we're told regular exercise can increase metabolic rate. Two people did 5 minutes of gentle exercise from rest, and we're shown the effect of the exercise on their heart rates, and we're asked to describe two differences in the response of person R and person S to the exercise. And this is that person R's heart rate rose more slowly than person S, and person's R heart rate leveled off, while person's S continued to increase. This usually indicates that person R is more healthy. For some reason, they don't try and hint at person R being more healthy. And then we're just asked to complete the line graph. Oh, we're asked to complete the line graph. I have not completed the line graph. I've actually just done the graph now. You simply take all the stuff from person S, put it down, and draw the label at the bottom and label its time in minutes. And then question 2.6. After 5 minutes of exercise, the heart rate of person S was 132 beats per minute. When person S rested, his heart rate decreased steadily at a rate of 12 beats every minute. Calculate how much time it would take for person S to return to its resting heart rate. Well, person S's resting heart rate is 72. No, it's 78. Whoopsies. So simply 132 minus 78, which doing quickly in my head is 54. Then you simply divide 54 by 12 to get 4.5. So it's 4.5 minutes. So it's quickly just showing why it's so important to actually check your working out. Otherwise I wouldn't have noticed that mistake. Okay, next up we have 2.7. The first 6 marker. A student made the following hypothesis about the heart rate of smokers and non-smokers during exercise. During exercise, the heart rate of smokers increases more than the heart rate of non-smokers, and we just have to design an investigation that will allow us to test this hypothesis. Well, first thing we need for investigation is two groups of people, the smokers and the non-smokers. So I started off by saying for the experiment you'll need two groups of people. Non-smokers and smokers, having at least 5 people in each group, and the reason we have 5 people in each group is just so there's enough people that there's not just one marathon runner who also happens to smoke who's really fit. Having the same amount of people in each group, making sure they're also the same gender, because different males and females have a different lung capacity that causes different increases in heart rate. And we get each person to do jumping jacks or any other exercise for 5 minutes, with them recording the heart rate for each person before and after exercise calculating the increase in heart rate for each person by subtracting their resting heart rate from the heart rate after exercise. And then you simply compare the groups to test the hypothesis. And while I don't actually state it in the question, and you're not meant to state it in the question, you would expect the smokers to have their heart rate increase more because their heart needs to pump more oxygen due to their lungs being less efficient. Now moving on to question 3. The circulatory system is composed of the blood vessels and the heart. Urea is transported in the blood plasma. Name 2 other substances transported in blood plasma, and it is carbon dioxide and glucose. It is not oxygen. I've seen people write oxygen for this question before. Oxygen is transported in the hemoglobin. 3.2. Some athletes train at a high altitude. Training at a high altitude increases the number of red blood cells per centimeter cubed of blood. Why is having more red blood cells per centimeter cubed of blood an advantage to an athlete? And this is due to there being more hemoglobin, which means more oxygen can be transported, allowing for more aerobic respiration to take place in muscle cells. And then for 3.3, which 2 blood vessels carry deoxygenated blood? The vena cava, which carries deoxygenated blood from the body to the heart, and the pulmonary artery, which carries deoxygenated blood from the heart to the lungs, with the pulmonary artery being the only artery to carry deoxygenated blood. Next up is shown 3 types of blood vessels. I've just labelled them for simplicity's sake. A is arteries, B is veins, C is capillaries. And we're asked which type of blood vessels carries blood into the right atrium, and that's veins that carry blood into the right atrium. You know this is a vein because the walls are a lot thinner because it's not having to deal with as much pressure. And that sort of links to 3.5, where I'm asked to compare the structure of an artery with the structure of a vein. And arteries have a thicker layer of muscle tissue than veins, and a thicker layer of elastic tissue than veins. And arteries do not have valves, while veins have valves. And the reason veins have valves is to prevent backflow due to the blood pressure in veins being lower, and the reason arteries have thicker walls is to allow for higher pressure blood to pass through it without causing damage. Next up, we're just asked to show on the heart where a pacemaker would be, and this is in the right atrium. I don't know how they would expect you to know this. I know this because in A levels I studied how this is where the SA node is, which starts the electrical impulse of a heart, so I knew it would make sense for a pacemaker to be there. I don't know how they expect GCSE biology students to know it. Maybe it's just something you get told to remember, but yeah, that's how I knew that. And what condition can be treated using a pacemaker, and that is an irregular heartbeat, or its more scientific name, arrhythmia. Then we're moving on to question 4, which is about chicken eggs. Student carried out an investigation using chicken eggs. They placed 5 eggs in acids for 24 hours to dissolve the eggshell, and then they measured and recorded the mass of each egg. Then they placed each egg in a separate beaker containing 200 centimeters cubed of distilled water, and after 20 minutes they removed the egg from the beaker and dried them gently with a paper towel, and were asked to measure and record the mass of each egg. And then we're simply shown a mass of egg without shell in grams and a mass of egg after 20 minutes in grams. And we're told to explain why. Well, firstly, do we agree with student that egg 4 was anomalous? And we're asked to give a reason why. And the results for egg 4 are anomalous due to the mass increase for egg 4 being less than the mass increase for the other eggs. And then we're asked to calculate the percentage change in the egg 3. Well, percentage change is final minus original over original, so it is 75.7 minus 72.4 over 72.4 times 100 to give it to you as a percentage instead of a decimal. So that's a percentage change in mass of 4.6. And then we're asked to explain why the masses of the eggs increased. And the mass increased as water entered the egg via osmosis from a dilute solution in the beaker to a more concentrated solution in the egg through a partially permeable membrane. Now, we're not explicitly told what the concentrated solution is. It could be sugar, it could be salt in the egg. The egg could have more sugar and salt in it. But because the water outside is distilled and pure, the concentration of everything in that water is obviously going to be 0 besides the actual water. So whatever liquid is in the egg obviously has a higher concentration, despite us not knowing what concentration they are actually talking about. Yeah. And for 4.4, we are asked to explain how the student can modify the investigation to determine the concentration of the solution inside each egg. And you can either write about sugar solutions or salt solutions. So you simply use 5 or more concentrations of sugar solutions, then plot percentage change in mass on a graph, and then determine the concentration where the line crosses the 0% change, with this being the concentration inside the egg. You probably would have done this with like either carrots or potatoes, and how it works is you simply have different concentrations listed on here, and then the percentage change of mass on the y-axis, and different concentrations listed on the x-axis, and you end up like drawing a graph, and the concentration inside the egg is where it crosses the x-axis. But we're not actually being asked to do that investigation, we're just being asked to calculate it. And then the chicken eggshells contain calcium. Calcium ions are moved from the shell into the cytoplasm of the egg, and we're shown information about the concentration of calcium ions in the eggshell and in the egg cytoplasm. And we're asked to explain how calcium ions are moved from the shell into the cytoplasm of the egg. Now, as you can see, in the shell it has a lower concentration, and in the cytoplasm it has a higher concentration, which means because it's moving from a low concentration to a higher concentration, it can't be via diffusion, and it has to be via active transport. So we simply write they moved from an area of low concentration to high concentration via active transport. And then it's a bit tricky, but you have to write down like the key characteristic of active transport, which is that it requires energy. Next up, we're moving on to question 5. Plants can be infected by fungi, viruses, and insects. Aphids are small insects that carry pathogens. Figure 6 shows an aphid feeding from a plant stem. I'm just showing a little nice diagram here. An aphid feeds by inserting its sharp mouthpiece into the stem of a plant. Give the reason why the mouthpiece contains a high concentration of dissolved sugars. And we need to work out what it's actually eating from the plant that would cause it to have a high sugar. And the thing that actually transports sugar in the plant's stem is the phlegm, or the phylum. I think it's pronounced phylum. And so the reason it has a high the mouthpiece has a high concentration is as the aphid is eating from the phylum. Next up, there's a little 5 marker. Plants infected with aphids may show symptoms of magnesium deficiency. Magnesium deficiency symptoms include yellow leaves and stunted growth. And we're asked to explain how a deficiency of magnesium could cause these symptoms. Well, this is mainly because magnesium, alongside other minerals, is needed to make chlorophyll, and the magnesium deficiency causes leaves to turn yellow due to a lack of chlorophyll, and therefore there is less light to be absorbed by the chlorophyll, because it's less efficient at absorbing light because there's less chlorophyll, causing a lower rate of photosynthesis, which leads to the plant making less sugar, causing the plant to convert less sugar to I don't even know what I wrote, to give less sugar to be converted into protein, stunting growth because they don't have enough protein. Next up for 5.3. A farmer thinks a potato crop is infected with potato virus Y. The farmer obtains a monoclonal antibody kit for potato virus Y. To make the monoclonal antibodies, a scientist first isolates the PVY protein from the virus. Describe how the scientist would use the protein to produce the PVY monoclonal antibody. And this is just, it's a 4 mark, it's always a 4 marker, it appears quite often, and it's just something you need to remember. That you inject the protein into a mouse, combining lymphocytes with tumour cells to create hybridoma cells. Then you find a hybridoma which makes the monoclonal antibody specific to PVY, and then the scientist clones the hybridoma to make many cells which produce the antibody. And it's just a very common thing, and the only thing that really changes whenever this question appears is the actual virus you're producing the antibody for. Question 6. Can't believe AQA thought this was a good idea. The question 6 was a cystic fibrosis question. They decided to they decided to have people write about how people with cystic fibrosis die young, and then they had them argue against the potential treatment for it. They had them argue about ethical concerns for a potential treatment. And obviously, because it's a genetic condition, people with cystic fibrosis were probably answering this paper. So basically, they had kids write about how they were going to die young, and then made them argue against the cure. Or what, not cure, a treatment. They had them argue against the treatment. And then obviously AQA had to remove that question, because people got mad. No kid should be forced to write about how they are going to die young and then argue against a treatment. Imagine you had a disease and then you had to write in your GCSEs you had to argue against a treatment for it. It's completely ridiculous. I can't believe AQA made people sad. Actually, I messed up my maths there. 300,000 divided by 3,000 is 100. But like, still, no kid should have to write about that. It's ridiculous. So unfortunately, I won't be able to answer because like the paper on the AQA website doesn't actually have it anymore. And like, if a teacher shouldn't be giving this out as mocks. Like, teachers should be following AQA guidance. I'm going on around here. I'm going to go back to question 7. It's the last question. Table 7 shows information about some food components in a cow's milk. How much more milk would a typical adult have to drink to get the RDA for calcium compared with the amount of milk needed to get the RDA for vitamin B12? First things first, we simply work out how much they need for calcium, which is 828.45 grams, and then we work out how much is needed for vitamin B12, and which is 266.67 grams, and then we simply minus one from the other to get 559.78 grams. And then this is a 6 marker. I don't know why it's formatted like this. PDFs don't like me. Describe how a student could test cow milk to show whether it contains protein and different types of carbohydrate. And first thing you'll notice is that this is a food test. So you simply need to state the food test for each thing and then show the result if positive. And that's like 2 marks for each food test. So for protein, use a burette reagent to test for protein by adding the burette reagent to milk solution, and it will turn from blue to lilac if positive. For starch, which is one type of carbohydrate, you use iodine test, and you add the iodine solution to the milk solution, which will turn from orange to black if positive. And then for the other type of carbohydrate, which is sugars, use Benedict's reagent to test for sugars, and you add Benedict's reagent to milk and boil the solution, and it will turn from blue to red if positive. One important thing is you need to actually state to boil the solution for Benedict's reagent. Other than that, it's pretty easy. It's 6 marks, but just you need to remember the food test. There's no real logic behind them, and it's just something you need to remember. And moving on to question 7.3. Firstly, you have a massive bit of exposition for what is seemingly a very tiny question, but it's used in 7.4 and 7.5 as well. Scientists investigated the effect of bile on the breakdown of fat in a sample of milk. The scientists used an indicator that is colourless in solutions with a pH lower than 10 and pink in solutions with a pH above 10. And then you literally give their method. Explain why the indicators in both tubes became colourless. Well, it's simply because by adding the bile, lipase breaks down the fat into fatty acids, with these fatty acids then lowering the pH, causing the pH to be below 10, causing it to turn from pink to colourless, as is stated up there. And in the last 2 questions, give the reason why the measurement of time taken for the indicator to become colourless might be inaccurate, and that's because the observation of the colour change is subjective. Like, some people may measure it as like when it turns partially colourless, some people may measure it as when there's like completely no hint of pink left, and there's sort of no way to tell it. That's why, like, in certain experiments, you use sort of like a light indicator to see, when you're testing something that becomes cloudy, use a light indicator to see how much light it lets pass through. and you say the results counts when only like 10% of light is being let through. And then for the last question, we're asked to explain the differences between the results in the two test tubes in table 9. And the two test tubes are the solution with bile and the solution without bile. And the solution with bile goes down quicker because the bile emulsifies fats, creating a larger surface area of fat, so the lipase can break down fat more quickly. Anyway, that's the end of the paper. I think it's a fairly nice one, besides the whole making kids write about how they're going to die. And I hope you enjoyed it, and I hope you found it helpful, and I hope you like and subscribe, and I'll see you next time.